The process begins with a PDMS microwell slide. Each slide consists of 16 individual arrays, each with 255 microwells. A solution containing about 25 to 26 cells is deposited into each array. This ratio helps ensure that only single cells settle into the microwells. Next, a lysis solution is added to the array to expose the DNA. A denaturing buffer is then added to destroy the chromatin structure and separate DNA into single strands. A neutralization buffer is added to neutralize the pH for amplification. The sample is now prepared for multiple displacement amplification, or MDA. A master mix of random primers, dNTPs, Phi-29 polymerase, and fluorescent markers are added to the microwell array. The arrays are then kept at a controlled temperature of 30 degrees Celsius for an overnight period. The primers bind to the DNA strands at random locations as the Phi-29 polymerase begins to synthesize the DNA. When the polymerase reaches a downstream primer location, it displaces the DNA strand, creating a new single-stranded DNA. Primers now bind to the new strands of DNA, and the polymerase begins to synthesize DNA on the branching strands as well. This process continues repeatedly, forming hyperbranching DNA structures and activating more of the fluorescent markers. The more DNA produced, the brighter the microwells will fluoresce. Images of each microwell array are captured every half hour by microscope to measure and track the progress of the amplification process. This provides the robotics system with the locations of each positively amplified microwell, so that the micropipette can be directed to each well for sample extraction, and deposit each sample into a PCR tube. A new micropipette is used for each subsequent amplicon extraction. An entire array can be processed overnight, yielding up to 400 individual single-cell amplifications. Each sample is individually stored in a PCR tube, and the DNA is converted into a bar-coded sequencing library to be sequenced in the next generation sequencer.